Okay, we start the program. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
Alhamdulillahirrabbilalamin. Nahmadu wa nusallimu ala rasul karim amma ba'du. Dear participant, many thanks for your gathering in our program in seminar with Professor Farahidah. Before the seminar start, we apa namanya, from my vice dean to apa namanya, opening this program. Time is yours, Mrs. Professor Nur Hasanah.
Oke, thank you Pak Arif. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Good morning to everyone, especially to Prof. Farahidah Mohamed. Yeah. Today is the 24th day of Ramadan. Yeah, hopefully, we all get the blessings and goodness of Ramadan. Yeah. In this session, I would like to express my gratitude to Professor Dr. Farahidah Mohamed, who available to share her expertise in the topic of the drug delivery design in this guest lecture. This guest lecture is one of a series of visiting professors program. Yeah. So we will follow by the next program with Prof. Farahidah in this program. This program arranged by Faculty of Pharmacy Universitas Ahmad Dahlan to increase the international activity and also international collaboration, especially between Kuliah of Pharmacy, International Islamic University of Malaysia, and Faculty of Pharmacy, Universitas Ahmad Dahlan. This guest lecture is arranged especially to our postgraduate students, masters and PhD students, but we also open to whom interest with this topic. So this guest lecture is open to all of participants who interest with this topic. Yeah. At the last, thank you very much to Professor Farahidah and to all our students and participants. Enjoy this webinar and you will get the latest information about the drug delivery design. Thank you. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you, Mrs. Prof. Nur Hasanah Mahfud. Before presenting from Professor Farahidah Mohamed, we will write the CV. I will write the CV. Bapak Ibu, sebelum kita mulai, saya akan bacakan terlebih dahulu CV dari Profesor Farahidah, guest lecture pada pagi hari ini. Beliau lulus farmasinya di tahun 98 di International Medical College dan 98 ya. 98 ya. Kemudian 99 apotekernya ya. Di University of Strathclyde dan PhD-nya di 2008 di universitas yang sama. Beliau sangat pengalaman di bidang drug delivery design. Ini beberapa pengalamannya sebagai farmasis di farmasis TNL farmasi di industri mungkin ya, di industri ya. Farmasis di Medilink Farmasi, kemudian terakhir di People Pharmasis di Hospital Muar Johor. Dan sekarang beliau di AIUM di Fakultas Kuliah of Farmasi. Demikian perkenalan beliau, banyak sekali grant-grant yang beliau dapatkan ini saya tidak sebutkan satu persatu, ini saking banyaknya gitu. Oke, untuk mempersingkat waktu, eh, kepada The Honorable Profesor Farahidah Mohamed, saya persilahkan untuk mengisi program kuliah tamu pada pagi hari ini. Silahkan, Prof.
Bismillahirrahmanirrahim. Alhamdulillah wassalatu wassalamu ala asyrafil anbiya'i wal mursalin wa ala alihi washabbihi ajma'in. Okay, I hope everyone can see my lecture slides. Yeah, okay, right?
Okay. Right.
Oke.
Alhamdulillah. So since this is guest lecture, I think I would do something similar to my other lectures to Kuliah Pharmacy students, whereby I will recite prayer of intention to study first before I start my lecture. Okay. So, Bismillahirrahmanirrahim. Alhamdulillah. Wassalatu wassalamu ala asyrafil anbiya'i wal mursalin wa ala alihi washabbihi ajma'in. Nawaitu ta'alluma wa ta'lim wa tadhakkura wa tadhkir wa naf'a wal intifa' wal ifadata wal istifada wal hatha 'ala tamassuki bi kitabillahi wa bi sunnati rasulillah sallallahu alaihi wasallam. Wa du'a ilal huda wa da'a ta'ala al khair ibti wa wajhila wa mardatihi wa qurbihi wa thawabihi subhanahu wa ta'ala. Amin ya rabbal alamin. Yeah, Alhamdulillah. The prayer intention to study just now was composed by our Habib Abdullah Al-Haddad, descendant of Prophet Muhammad SAW. So it's a very powerful dua. I hope everyone can continue to recite it before each class. So this is for my first lecture with UAD postgrad students, especially for the special program under the professor visiting program. The topic as requested by faculty UAD is drug delivery design. Okay, we go deeper and delve into what is drug delivery design. I wish that we can have same definition. The drug delivery is when what's constitute drug delivery. Okay, so for drug delivery, it is actually a dosage form to carry the drugs, because the drugs itself, they are very most of them is very bitter. They cannot just be taken like that, so it needs to be composed or designed into a suitable form so that it is palatable and convenient to be taken. Okay, and apart from the drug itself, it can, for simplicity, we call drug delivery design or drug delivery dosage form for all active ingredients that whether they are at R&D level, they are being marketed, already marketed. Okay, we call it as drug delivery system or DDS. Okay, it can be an advanced one, it can be a conventional one. So, and it also, apart from the active ingredients, where it can be from chemical synthesis, small molecular weight drugs, it can also be big molecules like protein, peptide-based drugs, a piece of DNA molecules, that is the active. The other components, we call it excipients that make up the whole convenient dosage form. The whole dosage form or drug delivery system that make it the drug to be convenient taken. Okay, so that is basically the drug delivery system. We need to study the drug itself. We need to study the material constituting the whole drug delivery system. And that will also include container closure system, which is the final packaging system. Because the final packaging system is also very important to ensure the whole formulation is stable throughout the shelf life. So that is basically the review system. Inshallah, we have a same terms, terminologies, definition on the drug review system. So for the outline of my lecture today, I'm going to touch on, maybe it's a revision, a timeline of drug development. Okay, and then I would like to share what are our responsibilities as Muslim drug designer. Okay, we are drug designer. we are Muslim. We have more obligations to fulfill. Okay, and I'm going to tackle based on Maqasid Syariah and SDG. Okay, SDG is Sustainable Development Goals. Okay, and then I will move on using and introducing again the concept of quality by design approach in drug delivery design. For today's topic, it's quite general. I will not delve into single components. I mean the thousands of materials out there on how to select, but these are a framework on how to start with the drug delivery design. Okay, and then I will give a case example of a simple drug delivery design, which is PCM, paracetamol plus honey suspension. So I will give the case example, a very simple one, drug delivery suspension. Okay. Right. So I'm sure everyone here knows the conventional dosage form, the drug delivery conventional forms can be from solutions, lotion, mixtures, creams, semi-solids, paste, ointments, suspension, immediate release capsule and tablets, and even powders. These are conventional. So these are the novel drug delivery systems. Okay, it can be from polymerics or inorganic materials and lipid-based materials. I'm sure everyone here is very familiar now with liposome, lipid nanoparticles, dendrimers and spheres, etc. So what we have to study is to study dual things. I'm talking about drug delivery. It can be even these small particles being compacted into a tablet or into capsule, depending on how we design them. Okay, so these are the novel drug delivery systems. But today I'm going to touch on the overall. Okay, these are the first thing. Just want to share the timeline of a drug development. If any of the PGs here, postgraduate students here, I believe some of them is in the field of extracting new compound from perhaps a plant, herbals, okay, herbs, so many plants, or even maybe animals, okay. So you are in this stage where it would require 3 years, perhaps, to identify the target molecules, okay. So these are active ingredients. You are still at a very, very early stage of drug delivery design. For this kind of research field or field area of interest, they are starting with extraction. So those who are starting with extraction, they need to think about, they are actually creating a new ingredients, a new active ingredients, where they also need to come up with a certain testing to build a birth certificate for the ingredients. So these are the first, yeah.
Hello, apa jenengan bukan Nino apa ya?
Ya, Ibu apa soalan? Any question? Or can I proceed? I think I can proceed. Okay. So that is the role of those who are doing extraction. Okay, they are building a new ingredients, they are testing, and they just need to make sure the extraction they produce from a plant which are consistently grow in a controlled environment as well. So starting from that extraction, we need to control the quality of the active ingredients so that it can be scaled up further. Okay, so that is extraction. And once we get the target compound, we test on high throughput screening. For example, you want to test what is the therapeutic effect of that extraction. So that is require other years, maybe another 3 years. Okay, and after we identify, perhaps it is effective to reduce blood sugar of diabetic rats, for example. These are preclinical development stage. Okay, And then we start formulating into a suitable drug delivery system, whether you want to make it as injectables products, or you want to make it as, if they are very stable, you already identify all the physical chemical properties of the active ingredients. We think it is very suitable for oral dosage form. Oral basically is the most convenient dosage form. Even though there is oral and transdermal is the most convenient. To me, my personal opinions is oral and transdermal. Inhalation, pulmonary on the other hand, although they give some of drugs, if appropriately designed, they can give a very good absorption at the lung area. Okay, but the way to take it, to administer this via either powder inhaler, dry powder inhaler, or metered dose inhaler, they are not convenient actually. For the patients, it's not convenient. So as much as possible, we need to create a compound that is stable and suitable to be formulated or designed into a topical, transdermal, or oral. This is the best, to me, the best and the most convenient. Okay, so for nanoparticles, even if you are in a nanoparticles designing of nanoparticles, you need to think of as much as possible to design them, to design the drug delivery system so that they are suitable for orally delivered drugs. Okay, so after that, after you formulate them and you already outsource and identify all the raw materials, critical raw materials attributes, okay, we now at the stage of scaling up, okay, and then we will apply for ethical approval and start phase 1 clinical trial. If these are a new drug entity, okay, NDA, a new drug NCE, sorry, new chemical entity. If it is a new chemical entity, it's going to be the first time testing in human. This will require a longer duration until it enter markets, before it enter markets. So we need phase 1 in normal subject, a small number of people, about 20 people, small one. These are the first trial in normal healthy volunteers. Okay, you will test whether they produce any severe side effect, and you will get into you will determine the maximum tolerable dose. So after that, if once you got the dose, you will close the study for phase 1. You apply for another phase 2, where you need to test on the actual patients. Okay, if it is intended for diabetic, you need to have a diabetic patients and test. And during phase 2, if the new drugs does not at the maximum tolerable dose obtained from phase 1, when you test in phase 2, they are not producing any therapeutic effect. They are unable to reduce the blood sugar level. So the new drug development will stop here. So these are the normal drug development timeline and the normal fate of the new drug development. So any drug that does not produce significant effect at phase 2, the drug development will stop there. It's basically failed. Okay. So if otherwise, we will test further in phase 3 multi-center randomized clinical trials. So once we establish the significant effect in the larger population, so the drug basically can be marketed, phase 3 will be closed and they will submit for registration. So within this phase, when we design the drug delivery system, we need to take care of all the quality, starting from the raw materials and all the quality of the manufacturing process as well. So that once you arrive at phase 3 clinical trials, this kind of quality material quality, the processing has been demonstrated as robust. So you can submit and ready for the documentation for submission for the regulatory registration. Okay, these are to help you to develop anything that you are in any stage here. You need to think of developing something meant for helping people, right? To give impact to human, impact in terms of saving their lives, because these are our role for pharmacists. For a designer in the drugs, we need to save lives. So these are our intention. So when we know these are the normal drug development process, we need to ensure we take care of the overall quality starting from the beginning. Okay. So in order to design the drug delivery system, we need to base on a certain principles. Since we are Muslims, we need to have a responsibility. We need to know what are our responsibilities as Muslim drug designer. Okay, I highlighted, I summarized into 4 points here. Number 1 is to give priority to common diseases. I mean, when we design certain drugs, drug delivery, of course we cannot do for all drugs, for all diseases, right? So our role is to need to do some prioritization. Okay, we need to give priority to common diseases. Okay, and perhaps that diseases, we need to find out what is the underlying cause of diseases. Maybe we have a very common one is hypertension, diabetes. But what actually underlying cause of diabetes, hypertension? It could be obesity, right? So what we need to actually prioritize is the creating a drug delivery meant to tackle obesity issue, something like that. And I'm giving ideas. And as Muslim, we need to be resourceful. Whether we are designer or Muslim traveler, we need to be resourceful. And in terms of drug designing, we need to integrate several sources. And this could be a quite latest discovery or construction using network pharmacology to arrive at a certain decision. Okay, I will show you a very simple example. And as a Muslim drug designer, and especially we are in the Malay archipelago, we maybe would like to narrow down our sources of ingredients to study or to dig more info, okay, or to verify. I suggest we also try to look at Malay medical manuscripts. Okay, if Chinese, they have Chinese pharmacopoeia, Materia Medica. Indian, they have Ayurvedic medicine. But Malay world, we need to give more ways to develop our own products, our own treatment, medicine from our own sources. So it's a very useful if we dig into Malay medical manuscripts. And then point number 2, our responsibility is to select the most appropriate route of drug administrations. Okay, we may choose the most convenient one, and because I'm talking to mostly postgraduate students, right? So I suggest to start with simple one before you go to sophisticated version. Okay, in drug designing. And then as Muslim drug designer, we need to be very particular about selecting the materials. Even when you do your extraction, if you are doing extraction, you need to ensure the raw materials that you use, the solvents, whatever ingredients that you want to put in to optimize your extraction procedures, you need to select them from halal materials. If let's say those involved in biotechnology, they need to do amplification of DNA of the gene using some media, using some serums, need to ensure that the serums or even the origin of the gene itself is from a slaughtered halal and slaughtered animals. Okay, It's not from POSAI. So please ensure this in your study or your research. Or even maybe we want to go for local first. Say for example, tackling or still extracting active ingredients from plants are still the best to go forward with new drug design. Okay, and then we need point number 4, we to ensure we are not wasting so much time and we are not using so much fund to develop drugs. Okay, I suggest it would be better for us to choose method of manufacturing of our drug delivery designs using existing manufacturing equipments rather than creating a new one which is so complicated and will need or require a lot of grants, a lot of funds to come up with a new machineries, for example. So better we go for existing technologies. There are so many to studies anyway. These are examples of Malay medical manuscript I'm talking about. There are some still available in this classical Jawi text, and some of them already transliterated to Roman, okay, to what do you call it? Alphabets, the one that we can read. Okay, so these are Malay medical manuscripts. One of the good sources, powerful sources, I could say, because they have so many. They have ingredients that is available. They are available in our Malay archipelago. Okay, so we can integrate the information given here with using several network pharmacology tools. I'm going to show you what is network pharmacology. This network pharmacology is an interdisciplinary field that studies the interactions between drugs and biological networks. We have studied the drugs or medicines. They exist, I think, already hundreds of years. And with the advancement of so many AI systems or AI technologies, they can come up with various software and produce various links here and there. Okay, so these are bioinformatics. They already have a certain database and they have other technology to do the docking, for example, a computer aided system, computer drug aided design, something like that. So everything is being compiled and they come up with the term network pharmacology, where it challenged the traditional one target, one drug. Usually, what we understood with the single compound, the target of pharmacological reaction would be only one side of one side of receptor, one receptor side to produce one pharmacological action. But with the network pharmacology available, it challenged this traditional concept, and we can explore potential of multi-target drugs. It can be one drug can target various one drug or multiple drugs together being composed or being designed into a one single drug delivery system taken together and produce a multiple effects. Is also possible depending on type of drugs you are looking and work with. So one tools that network pharmacology employs is MEI, metabolic biomarkers enzyme interactions. So it is I could say powerful software to play around with to check even what is our compound and possible compound metabolites interaction, their interaction with biological systems. Okay, so everyone now can try it. So these are another site that already have the database, the listing of so many ingredients. It is called the Human Metabolome Database, HMDB. Okay, you can try after this to browse and search your own compound. For example, if I'm working with chrysin, then I can check chrysin metabolites. There will be a few results of the search. You can check on it. Okay, and just now I mentioned about our responsibility to prioritize common diseases, right? If you want to check what are common diseases throughout the world, you can go to A to Z list of common illnesses and diseases here at the website, and then you can check it. And of course, we need to do counter checking or cross reference with WHO website, for example, to check on the prevalence of that common diseases. So you can start by prioritizing what you want to study, what you want to design for the next drug delivery system. Then what we are doing now actually to fulfill Maqasid Syariah, whereby you want to safeguard the especially the life here. Maqasid Syariah here basically is to safeguard 5 values: life, lineage, faith, intellect, and property. And pharmacy, especially Muslim drug designer, we intend to design drugs to save lives. Okay, saving the lives could be through two methods. One is to prevent disease. Second one is to treat the disease. So these are also saving life. One, maybe one or two of you is concentrating on preventing disease, creating something, delivery system to prevent disease, where we can actually safely say it is a nutraceuticals products to prevent diseases. If in medicine terms is being used to treat diseases. So whatever it is, we want to safeguard the life of our Muslim, of our populations, human populations. So this Maqasid Sharia concept, actually they are very in line with Sustainable Development Goals promulgated by WHO. Okay, and in terms of saving the life, it would be more appropriate to align them with the SDG goal number 3, to have a health and well-being. Okay. And of course, in doing that, we need some innovation. Innovation would be innovation here. Actually, innovation. Innovation, industry innovation and infrastructure. Sorry. We are at here. And we need a partnership for the goals. So for today, maybe we can start do some partnership with UAD. UAD will have SAM, XRD. Perhaps we can share these facilities, okay? And maybe here we have more on manuscript, medical manuscripts, we can share with you. So that we have more resources, human resources to work together. So these are basically our goal in designing a drug delivery system. And one more thing, as a Muslim drug designer, we maybe these are the concept that everyone here can hold up to. Halalan, Toyiban, creating drug delivery design that is halalan and toyib, and wise medicine. It's a wise medicine, okay? Or wise nutraceutical. They have the attributes of wholesomeness, they have the attributes of innovation and sustainable and synergistic, and in terms of equitable, they can be accessible by any levels of populations. So basically, these are the 3 in line with SDG, 3 SDG: good health, industry innovation, and partnership for the goals. Right. So when we are talking about role of a Muslim drug designer just now, we need to have some prior knowledge, and after we know what to prioritize, we need to also have prior knowledge on drug design, especially on route of drug administration. Okay, so I have few slides talking about these routes of drug administrations, which I think most of you already know about this, or they knew about this, or they aware, and these are just a very basic revision. Okay, so route of drug administration is divided into oral, enteral route, or parenteral route, and other routes. Basically divided into 3: enteral, parenteral, and other routes. So for enteral routes is the most convenient, cost-effective, and most commonly used medication administration route. And when we formulate into oral drugs, the way the storage condition usually applies to these orally delivered drugs, okay, is actually low cost. It's just we just need room temperature to store them, okay, because they are oral, they are usually solid, they are dry state, very low risk to be contaminated with microorganisms, and if they are dry. If you are formulating into suspension, even liquid form, they have a certain requirement for storage, but not as heavy as the requirement for injectables. So those of you using or who are working on nanoparticles, usually they are also very sensitive materials, hygroscopic. It needs you need to think of how to tackle the issue of stability. Okay, maybe you're using a certain packaging system, special one that can sustain and preserve the nanoparticles at room temperature. When we design drug delivery system, we need to also know the storage temperature is very important. Otherwise, if your drugs is not stable, the whole delivery system is not stable, or even they are stable but only at a cold temperature, this will incur another cost. So not really a wise delivery design. Okay, drug delivery. So if you are designing orally delivered drugs, we need to ensure a few things lah. Prior knowledge like what is the absorption like in small intestine, okay, and the bioavailability. Of course, it will rooted back to the physical chemical properties of the ingredients, and need to ensure the first pass effect is tackled. So when you design the drugs, you need to give a appropriate strength to one dosage unit, counting the portion of the possible first pass effect that will be encountered by the drugs when taken orally. Okay. The other enteral route is sublingual. Okay, sublingual or buccal route is another form of enteral route, where this one can bypass the first pass effect. So when it bypass first pass effect, when we design the drug, we can give a lower amount of the active ingredients. When we cut the concentration or the strength of the active ingredients, we can also cut the cost of medicines. Okay, so these are the things that when we design drugs, we also need to think of. And I'm sure all of you already know why this drug need to be delivered by a sublingual or buccal is to give to get a very fast effect of the drugs. Okay, and when compared to sublingual tissue, which has highly permeable mucosa with rapid access to the underlying capillaries, buccal tissue is less permeable and has lower drug absorption. So these are just an important points that we need to know about route of drug administration when designing drug delivery. Okay, another enteral route is rectal route. Even though these are part of the organs in human that can deliver drugs, but of course, if any one of you have ever administered drug via rectal, you know it is very inconvenient. Okay, so that's why when I talk about when we want to design, we need to think about route of the administration. Okay, and we can prioritize the easiest one. Even for the rectal for manufacturing, they are having a quite a more sophisticated environment when you want to manufacture this kind of rectal delivered drugs, rectally delivered drugs. Okay, moving on to other route of drug administration known as parental routes. This usually the products, the end products, the finished product will require a special storage condition. Okay, very rare that we see the products of injectable products require only room temperature, unless they are formulated into a dried stage that you need to reconstitute with some solvent or water for injections. So this kind of products, IV, particularly for protein or peptide-based drugs, RNA or DNA drugs, we need a special storage condition. So whatever it is, we need to study where you want to deliver, what is the fate of drug administration. Okay. Right. Other parenteral route is intramuscular muscles, and injecting the drugs at the muscles, through the muscles, and usually intended for sustained release effect. Other parenteral route is subcutaneous. Okay. Subcutaneous tissue has few blood vessels, therefore the medications injected undergo also slow and sustained rate, because the diffusion of drugs taking place slower than IV, than through IV. Okay. Other route of parenteral is intra-arterial. Okay, although these are not commonly used for drug administration, some of you may be involved in creating contrast materials. Okay, so you need to study what is the contrast material, what is the requirement of this contrast material when injected into intra-arterial. So these routes is usually used to map and know the regional chemotherapeutic agents for cancer treatment. Okay, other routes of drug administration is by transnasal delivery. Can be in semi-solid, can be just a solution you spray on your nasal, and can be transdermal, can be pulmonary and vaginal. So we need to prioritize which one. We need to study the routes. So these are overall routes when you want to create your drug delivery system. Okay, enteral. These are just to summarize: parenteral, topical, inhalation. So to me, my personal opinion, the oral and topical is the easiest one to administer drugs. So for those from work with nanoparticles or the more next generation particles, these are the common routes of drug administration. It can also be formulated to oral routes. You need to take care of the stability of your nanoparticles. It can also be administered by dry powder inhalation, and can be formulated into nasal and transdermal. Ocular delivery also popular, but I don't think ocular delivery is convenient as well. I myself, even I didn't wear any contact lens. Alhamdulillah, so far my eyesight is okay. But I think it's very, very sensitive lah, eye, as a point of route of drug administration. And IV is there's no other suitable routes. IV is the easiest to inject and give the drugs directly to the bloodstream for fast effect. Actually, what you need to know about these routes. Okay, for the design of drug delivery, these are the important things. We need to study the structure and function of the organ related to the routes of drug administration. We need to study the fate of drugs administered via the routes. Say, for example, if you choose oral, you need to study what happened to oral delivered drugs. After you consume the drugs, okay, they will arrive at the stomach, they will arrive down to the GIT. Stomach, what will they encounter? They will encounter enzymes, so many acids, all the gases. So what will happen to your drugs at the end of the day? So these are the fate of the drugs that you need to study. And then you need to ensure you know what is the percentage of drug absorbed and metabolized upon administration via each of the routes that you're interested in. And about the route of administration, when you design the drugs, you need to know the specification for the drug candidate suitable for the intended route. Okay, you say, for example, you want to design drugs by liposome, but your active ingredient that you want to work with is two-way big. You need to do something to ensure it can be administered via the skin. Okay. Right. So moving to the next section for today's lecture is talking about this quality by design approach in drug delivery design. Okay, I'm talking about the quality here. So what actually quality that we want to look into? Okay, and this quality by design, there is a specific guidance documents. We call it guidance document. Okay, it's a guideline which is composed by a committee these committee members. They are experts in this field, so they come up with a guidance and guideline that we can use, especially for industry. I'm talking about industry because at the end of the day, when you create the drugs in the lab, our intention is to our intention or our hope is that this drug that we create in the lab can finally enter market, right? Can finally give impact to you, to maybe prevent disease or cure diseases of human. So you want to save the lives, and if we want, as mentioned in I think surah, what is the surah? If we save one life, it as if we save the lives of the whole people. Okay, so and talking about the quality again, about the raw material, the responsibility of us as a Muslim drug designer, we need to take care of the halal, right? So the halal parts is also an obligation. When we there is one hadith, strong hadith, I forgot to insert here. Hadith from Anas, if I'm not mistaken, narrated that Nabi Muhammad SAW, Prophet Muhammad SAW mentioned, if you want to, if we have certain disease, we need to, it is compulsory for us to find halal treatment for the disease, okay, and not haram. So this hadith is very strong. It gives us to message. Hello? Hello? Anyone want to ask anything? Tiada?
Nantinya, Prof.
Okay. So that hadith mentioned two strong messages. Number one, if you are sick, it is obligatory for you to find the treatment. Obligatory to find treatment. You just cannot sit and just wait for the disease to cure by itself. It is one way of treatment, of course, but it depends on the disease. And when you are finding that kind of treatment or medicines, you need to find halal medicines. Okay, so as Muslim designer, we need to provide halal medicines to our Muslim patients, so that there are alternatives for the Muslim patients to take and cure themselves. Okay, so these are our obligations as Muslim drug designer. So what quality we are talking about when creating or designing drug delivery systems? These are based on, again, ICH harmonized pharmaceutical development Q8R2. There are a few Q numbers, R1, R2. For this one, pharmaceutical development is Q8R2. Okay. And this ICH, International Conference of Harmonization of Technical Requirements for Registration of Pharmaceuticals for Human Use. Okay. When we are doing the design of drugs, our intention is to register, finally able to register our product so that it can enter market, can give impact to humans. Okay. So that's why starting from the lab stage, we need to try, understand, and try to develop our or create our drug delivery based on this guidance document, pharmaceutical development QR-R2. So inshallah, when we do this kind of development, following these guidance documents, we are not wasting our time. We are doing appropriately according to the regulatory requirement, to fulfill regulatory requirements. So inshallah, it can finally enter market, inshallah. Okay. how to get rid of this? Okay, it's okay. Right, these are table of contents in the pharmaceutical development Q8R2. As you can see here, they mentioned about components of the drug product, where I already mentioned about our active, whether they are drug or whether they are natural extracts. You can check and develop or design your drug delivery according to this document. Okay, drug substance, and the major parts in drug delivery system is excipients. Okay, whether they can be from generally recognized as safe ingredients. Okay, usually from that list ingredients, which are so many, can be from polymer, cellulose base, etc., etc. And then the drug product itself, how to do the formulation development. And when you do formulation development, you need to also ensure you put in your formulation, when you do certain percentages of your formulation, right? You need to have certain overages. So overages is important to ensure the active ingredients has some reserve according to the claim percentage of your active ingredients. Say for example, I am making 5% methyl salicylate ointment. Okay. And the way we manufacture the ointment will have a certain waste of raw materials. After it solidifies, the waste will be stuck in the vessel, mixing vessels. So we already know a certain percentage. We already check how much is the waste. So that how much is the waste is we will recalculate the formation by having additional overages here for the active ingredients as well as certain excipients. Okay. So these are what we meant why overages is important. Because at the end of the QC control later on, you need to quantify the 5% methyl salicylate, for example, methyl salicylate as the active ingredients. Whether this tube contain 5% of methyl salicylate or much less than below the specification. So overages will give an advantage to the for us to meet the specification. And when some of the processing methods involve heat, right? When you heat something, of course, some materials will get lost to the environment or will degrade. So because if you are at the lab scale, you need to check everything. How much is being degraded when you heat, or how much from the theoretical, the first stage of weighing all the raw materials, at the end of the process, when you get the nanoparticles, for example, in the dried form, how much actually is left. So these are the left one, you actually need to cover the certain percentage with overages. Okay. So that's why when you do the drug formulation, you need to ensure you know every single steps, and you know that every single step will have certain impact on your active ingredients. Okay. So you need to ensure you have appropriate reserve how much to do the overages. And of course, everything, every drug products, even the raw material itself, we need to know very well what are the physicochemical properties. If the drug, what are the biological properties of the drug? And when we do the lab scale formulation, of course, you are now using lab tools. They are very different. When you want to do the scale up, you use a machine, okay? Even to prepare a simple cream at cream lab levels, preparing creams very simple. You can just dissolve, or you can melt certain excipients in using microwave, and then you just mix with even blender. Okay, but and that blender has a very different diameter, very different mechanism of mixing as compared to the machine when you do the scale up. So when you prepare at lab level, prepare your drug delivery particles, whatever it is, you need to try to imagine what kind of machine that will be involved during the scale ups, and what kind of things or machine that you can revamp to make sure the process at lab scale, sometimes the process at lab scales or majority process at the lab scales, they have a very they have a lot of drawbacks. Okay, we cannot close properly. There are a lot of volta solvent going out. But at the labs level, at the scale up level, this will be taken by appropriate design of machines. So you need to also dig more info what kind of machines available when you want to do the scale up, even if you are still at the lab scale. So one more thing is container closure system is being emphasized in the guidance, because as I mentioned earlier in my lecture, stability of your finished product is very important. It comes not only with drug delivery that you formulate at the lab scale, just the nanoparticles, the dried one, after you maybe freeze-dried them, you got the final, and you store. You need to ensure the way you store with appropriate container closure system to ensure when you do the further testing of your nanoparticles, they are not degraded and they are not interacting with the container itself. There are a few instances where when we do gamma irradiation to sterilize our nanoparticles or to sterilize our products, they get they degraded. Okay, and they interact with the container itself. So make sure the container you use does not interact with your drug delivery that you already formulated, whether they are in nanoparticles form, semi-solid, creams, lotions, okay? And then the other things which is important to take care is the microbiological attributes of your formulation, of your drug delivery, because this will be part of the stability study. How much it can sustain or prevent contamination, the growth of any contaminants. Usually the contaminants we are talking about here is the microorganisms. Yeah. Okay, so these are the things need to study before it can be a final finished product. And then we need to study the compatibility. Okay, in this guidance, they put compatibility at the below parts. To me, if I were to make a guidance for human, I will put compatibility at the up parts here, to ensure that every single ingredients that we use from the drug itself and the excipients, they are compatible. I will show you some example. Okay, this page is showing you table of content still at part 2. Okay, if you get access, you can get access. Just click and find ICHQ8R2. You will get access freely. You can download and read it and try to understand it. Okay, so part 2, at the part 2, annex to pharmaceutical development, which are very important. Okay, you need to know these are the elements of QBD, quality by design. These are the elements of pharmaceutical development based on quality by design. The elements are number 2.1 until 2.6. Okay, you need to have the quality target product profile. What is it? We need I will explain. You need to know what is critical quality attributes. Okay, you need to know the risk assessment. And I believe some of you are already familiar with design space. If you're familiar with response space methodology or factorial designs, these are kind of design space they are mentioned here. Okay. And then control strategy, how to control each of the raw materials and also the finished products. Okay, and the control of the machineries. And finally, the product lifecycle management and continual improvement. So these are all parts of quality by design. We need to design the drugs delivery by ensuring the ingredients we put in a quality ingredients, a quality process. Everything is being controlled so that we can actually produce a consistent product quality. These are pharmaceutical. It needs to be a quality, efficacious, and safe. That quality I'm talking about. Okay. So to give you a different understanding on the key elements in QBD, okay, quality by design. These are what we want. We want the QTPP, quality target product profile, that identifies or links to our critical quality attributes, CQA. And we need to do the product design and understanding the drug delivery system, including identification of critical material attributes. So each of the material you use, whether it's starting from your extraction, amplification of DNA, for example, the serum, the salt, the water, the bacteria, antibiotics that you use, or the starting passage numbers of your cells. Okay, these are critical material attributes you need to list out. Okay, these are all done at lab scales. You need to understand this. So the next, after you have done everything, you have identified what is your critical quality attributes, so that you produce every time you produce is consistent results. Next is to design the process. Process design is actually the scale up process using machineries. In the lab scale, maybe the most you can produce is 1 kilogram. Further than that, you may need a scale up using a certain machine. Okay, some scale up can be a pilot scale up where you can produce 10 kilo, 5 kilograms, even 3 kilograms. These are still pilot scale up. But it can some of the manufacturing or of dosage form, they already created a lab scale that managed to produce 3 kilos, 2 kilogram even. Okay. So process design is to understand, it is part of the quality by design, is to understand including identification of critical process parameters, CPP, that link the CMA and CPP, that link, integrate what are their interaction between the critical process parameter and critical material attributes to the CQA, critical quality attributes. And then we need to put in what is the control strategy that includes spec for the drug substance, the excipients, and drug product, as well as controls for each step of manufacturing process. These are part of GMP. And we need to study the process capability. Usually we study this based on 3 consecutive batches that we do. We call it process verification or process qualification. Okay. The term is big under the term process validation. But under process validation, there are a few process qualification and verification need to be done first. So process capability is one, and continuous improvement are parts of QBD, and they are GMP. And under GMP, we need to know what kind of tools to use. Prior knowledge is very important, where you can get you actually already obtain it from your undergraduate studies and from reading many articles. Okay, these are prior knowledge. And how to do this assessment? Need to know simple tools to do this assessment, design or experiments, data analysis, and PET, process ethical technology. These are elements in or tools used in quality by design. We want to design a quality medicines, maybe the medicines meant for prevention or disease treatment. Okay, but you need to put in the quality in your design. So I'm going to go deeper for part 2 and okay, say for example, these are case example already to illustrate what this guidance are requesting us. Okay, say for example, I'm sharing a development of a fusion drug. Case example, we developed these drugs, paracetamol honey suspension. The name of it is simple, just suspension. Okay, but it's a PhD work. Okay, and we managed to register this product, get clearance from the regulatory. Because there are certain, when it comes to regulatory requirement, there are certain things that we can tweak, not to tweak in terms of, we know that honey can be excipient, so we just declare it as excipients rather than active ingredients. That will make the overall regulatory requirement easier, okay, easier to register. But first of all, we need to know what are the kind of regulatory requirements needed, and so that when we develop the design of suspension here, we will develop them according to the regulatory requirement. So first of all, we need to do the compatibility study between the API here, paracetamol, everyone know, acetaminophen, then the different name, okay, and or active ingredients. If you use honey, we want to declare honey, if you want to declare. Otherwise, we just regard honey as excipients. So this paracetamol honey suspension, API is the paracetamol. The excipients are a few things: preservative, the stabilizer, the pH regulator, and the honey itself as the solubilizer, for example, and the taste masking. Okay, excipients. So API combined with these excipients, what is there any interaction or not? So we need to do the compatibility study. The easiest is to conduct this ATR or FTIR. Okay. Okay, and then we need to create quality target product profile. These are based on the guidance document ICHQ8 that I showed you earlier. And then we need to list the critical quality attributes associated with suspension, associated with your drug dosage form. Okay, so you need to list the critical quality attributes. If it is a tablet, critical quality attributes will be the hardness of the tablet, friability, the dissolution test, and the dissolution time. Okay, that is the CQA for tablet. For suspension, we will show it later. And then you need to do the risk assessment to link material attributes and critical processing parameters against their impact against CQA. Okay, so this risk assessment is actually an important step, because otherwise you need to do so many experiments to know what is the impact of excipient A to the processing parameter, or what is the impact of processing parameter mixing time, or mixing steps, mixing procedures, whatever it is, to the material attributes. There will be so many. So what you need to do is to do the ranking, do the risk assessment based on your prior knowledge. Prior knowledge, I mentioned, you already got it from undergrad experience, undergrad teaching learning, or you can also now at postgrad levels, you need to read a lot more of articles using the same or working with the same ingredients. So you need to know what other people's finding. Okay, so these are your prior knowledge. And in developing a drug delivery system, after that you need to know how to analyze your active ingredient, how to analyze paracetamol from paracetamol honey suspension. So basically, these are developing and take a method to analyze the active ingredients, or API. If it is paracetamol, well-established drugs, the analytical method for paracetamol suspension is there in USPBP. What we only need to do is to do verification. Okay, there are certain parameters that can be waived as compared to a new validation protocol. So after you develop analytical method, you need to validate the analytical method, and then after that you will think about how to scale up. So these are the designing of the process to scale up. Okay, when you design the process, of course, you also need to know your prior knowledge, use your prior knowledge. Okay, say for example, you know that suspension is just need a simple machine to mix them. but you may have to provide a few small machines to dissolve a certain materials first before you put them into a main mixing tank. So these are suspension manufacturing process in the plant, in the factory. So you know these are prior knowledge, so you can design, okay? And then you need to validate the process and you need to conduct stability study. Okay, the best to conduct stability study actually after you manage to produce anti-radiation step. But it is very very helpful if you can conduct stability study at your lab scale. This usually what we also do because you want to check what is the effect of temperature, effect of humidity, for example, to get a preliminary data on our product stability. Basically, for the case example here, these are the flow. First of all, you do preformation competitive study as mentioned in the ICH guidance just now. They need competitive study. And you need to check with your regulatory side what is whether they also part of the requirement to submit data on competitive study. In Malaysia, our drug regulatory registration, yes, we do need to submit competitive study data. Okay, talking about the competitive study, we can do and use the SC or ATR and the ATR to check. These are the two equipment that is less invasive or they can preserve the raw materials. They do not use any huge amount of heat to degrade the raw materials. Otherwise, the results will not valid. Okay. And then after we have the competitive study, if the ingredients of our choice based on prior knowledge, we selected a few ingredients, we need to formulate and do the characterization. Talking about this case example is paracetamol honey suspension. We will characterize based on characteristic of suspension that we need to characterize, which are particle size distribution based on the PDI value, zeta potential, which give the which reflect the stability of suspension, the viscosity, okay, pH, and we need to do some animal study to check whether our product has certain superior effect to the existing paracetamol in the market. In terms of viscosity here, okay, of course there is no value that we can be based on. What you need to do is to compare with existing paracetamol suspension in the market, already acceptable by patients, already in terms of user experience. The user can be it can be easily poured into a spoon and then it can be taken. So that kind of reference product, we can check their viscosity. So we can put in our specification of their viscosity to our viscosity here. Okay, the specification. So this is how we develop the paracetamol suspension. And then we do anti-chemical verification. In paracetamol formulation, we need to do two things. Not only active ingredients, anti-chemical method, but also the degradation, the impurities, which is for aminophenol, for paracetamol. How to know what kind of impurities you need to do is to go to USPVP, you check under paracetamol monograph, and they will mention these are kind of impurities you need to check. So after we formulate into honey suspension, of course, there are other excipients coming into the paracetamol, right? And there are suspension. There is water inside. They can degrade paracetamol. But how much degradation will take place? The degradation, the level of impurities need to be controlled up to your specified stability shelf life. Okay. So that's why we need to also do anti-chemical method, not only paracetamol, but also the alpha for aminophenol, the impurity of that paracetamol. And for our products, the development, alhamdulillah, after scale up, after we managed to tackle all the issue at the lab scale, find what is the best preservative, what is the best stabilizers. Okay, we select them and do the scale up process, design of the process. So again, the design of the process, we need to check the appearance, assay. Assay is being done by using the Antico method we validated here. We need to check, say 10 bottles, what is the uniformity of content of paracetamol in that 10 bottles. So these are the uniformity of content. And then we need to check based on the requirement for suspension. There is one requirement that is to check the deliverable volume. Okay, so that if you are working with tablets, you are working with creams, you need to check what are the pharmacopoeia requirement for the dosage form. If injectables, what is the other extra additional requirements? So you need to check that. So for the scale up, using a big mixing tank, what is the particle size after you do the process? What is the, again, the zeta, viscosity, pitch, and do the stability. Okay. Right. So this doing the compatibility using FTIR just now. API plus excipients, we do the rapid screening using DSC, and we check whether the DSC profile thermogram, is there any shift or not between the API alone and API plus excipients. Okay, so this is how we check. Either using DSC or usually we use both because want to back up. We want to make more evidence. Okay, we use FAR for the single compound, single materials against the mixtures. Okay, so we will compare the mixtures profiling, and fingerprint and the thermogram from the DSC data against the single materials. So if they are okay, we can use it. If not, you can do further study whether you want to check what is the incompatibility issue, or did they decompose into what kind of things, you can check if you have extra fund. Otherwise, you just move forward, use a different ingredient. So these are example of what we have done, the FAR data, honey plus what is this? That's one of the materials we put in. Honey and another materials, honey and another materials, and honey and PCM. So we combine all the FTR FTR spectra here with the honey alone. So we check whether they have interaction or not. So from here, the all ingredients that we want to use in our suspension, as and especially with PCM, compared with honey, there is no degradation of honey using with all these materials. Okay. Even though the certain region of fingerprint region here, there is certain different in lengths, we need to find out from whether it is belong to PCM or interaction, what is happening here. So from here, we can safely say that there is no interaction. Okay. The easiest one, if you are actually using very common ingredients available in the market and already being used in so many dosage forms, the easiest is to check this book, Handbook of Pharmaceutical Excipients. They have the compatibility informations between drug A and excipients A, for example. Okay, so they have highlighted and compiled what people have done, compiled in this book, and you can check. If you already check here, you don't really need to do the FTIR or DSC because you have already a reference book that mention your ingredient A do not interact with ingredient excipient A. So these are just words and statement you need to produce when you submit your document for registration. under the compatibility topics. Okay. Okay. All right. For the paracetamol honey suspension, we also study the materials attributes. It is actually a quite simple exercise. As many of the ingredients are not really sophisticated ingredients or not a rare ingredients. Okay, they are very commonly used. So what we need to do is to check with manufacturer the COA, Certificate of Analysis, for each raw materials. And we need to compare the specification stated in the COA with compendial. If we usually here in Malaysia we use USP or BP lah. Okay, Indonesia they have Indonesia Pharmacopoeia, so you can also use this. And when we develop our drug delivery system, we as much as possible incorporate this halalness as one of your specification. So how to verify a certain raw materials are halal? This need another lectures. Okay. I'm not going to touch it here. Maybe inshallah during the visit to Georgia in May, inshallah. So I can also give a lecture on halal. And then we need to conduct testing of raw material testing, okay, based on pharmacopoeia to verify the quality. So these are how we control the quality and based on the quality by design. I managed to get this Indonesia e-pharmaco, how they pronounce it. So these are Indonesian. So you can also use this to check your raw materials testing. Okay. I've been mentioning on the risk assessment where we need a prior knowledge to do it. Okay. Prior knowledge you can get from you already got it from your undergraduate exposure as well as now you can read more. And you can also do this exercise with your supervisors. Okay. So you can rank the most critical processing parameters. Say in the paracetamol honey suspension, we have listed the documents. Start with the what 4M 1E. 4M. 1M is material, man, machine. One more is what? Forgot. And E is environment. So these are the things that we listed down. The machine, what kind of impact of the processing that have impact on the CQA, called critical quality attributes. Here for the suspension, critical quality attributes will include zeta potential, particle size, humidity of content, and others like PDI just now and viscosity. Okay, so that is CQA for suspension. Okay, and then we listed down what kind of processing parameters that can have impact on this CQA of suspension. So we found out that one of the equipment that we need to use is milling, a colloid mill. Okay. So whether how much colloid mill will have impact, that will be based on prior knowledge. We need to read more. And whether the mixing tank has any effect or not, we also need to read more. Alternatively, if you are working with a well-established company, factory, right? They already know their operators, their technician, at the production site, they should already been exposed to many products type, and they actually can give you input on what kind of critical processing parameters for this mixing tank to produce suspension, for example. Okay. Or if you are making tablet, what kind of processing parameters? Say for example, you are using granulators, what kind of processing parameters of granulators can have impact on the CQA of the tablets. For example, if you are making a tablet. Okay. We mentioned about QTPP just now, one of the mentioned in the guidance. Okay. Back to paracetamol honey suspension just now. We have created our QTPP profile. We have listed down route of administration, oral, therapeutic category, analgesic, antipyretic, and dosage form we want to do is suspension. Active ingredients, we already identify paracetamol and perhaps honey. If you want to declare as honey as active ingredients. Otherwise, you can just shift the honey, move to excipients. And then excipients. Okay, we during our creating first phase of the QTPP, we may we perhaps don't really have we don't select the stabilizer that we want to put in. We are yet to select preservatives and pH regulators. There are so many, right? But we know from prior knowledge there are certain stabilizers that we want to test, the best one. Usually we to simplify, we may choose 5 stabilizers that want to test, or even 3. And then preservative, we already know that the common preservatives used in common preparation of paracetamol in the market is metaparaben, is propylparaben together. Very rare to see only one, because each of the preservative will kill a different microorganisms. So we need both. So for preservative, maybe you want to test other preservative which are paraben free. That is up to you. When you want to design your drug delivery system, you need to just if you want to choose them, you need to do some preservative efficacy test. Okay. So for first phase of creating QTPP, this are what you can be based on. Great. And then the rest, you need to select them from the halal criteria and also from the doing the testing at the lab scale. Example, selection of ingredients based on testings. Okay, we want to select what kind of preservative we want to put in. We check the impact of pH of the suspension. And we also want to check the type of stabilizers. Different stabilizers at different concentration, they produce different stability based on zeta potential. Okay, so we also test that. And finally, we get how much we want to put in and what is the best stabilizers in our drug delivery formulation. In this case, suspension. Okay. Okay, these are actually a table just to share with you. This is a kind of table that we produce. We need to keep in check whether we produce enough or sufficient or not before we want to submit for registration. So for example here, okay, just for example, I just take one for example here, raw material testing. If we have 6 ingredients, we have 6 total documents. Which include the standard testing protocol for the raw material A, for the raw material 1 until 6 raw materials. Okay, these are example of raw materials. They are important because these are where we control the materials attributes, CRM, CMA, sorry. Example. So for analytical method validation just now, we also has a few documents to prepare. Okay, AM report. Okay. For microorganism or preservative just now, one important thing for all products is to have this microbial contamination test validation. We need to produce a certainty and evidence that our products is not being contaminated throughout the shelf life. Or it can sustain a certain microbial limit. Okay, so we need to validate the testing and produce the data. So these are applicable to all products. And if we are not sure about the microorganism, how to do the test, etc., we need to seek our some help from those microbiology specialists. Some terminologies used is quite new to us, so you need to know a few terms when you do the MCT validation. Right, and until at the end, we will have about 36 documents to be prepared before we can submit for registration. Okay, these are at Malaysia regulatory requirement. when you do the drug delivery development, you can also check here, for example, additional testing needed. If you are producing a formulation meant for dry powder inhaler, you can check at USPVP what kind of additional testing required for dry powder inhaler. If suspension, you just search for suspension. Okay, you can go and check. And for this one is example of an active ingredients, paracetamol. I will find paracetamol monograph. These are another example of a drug, oseltamivir, antiviral drugs. We can check it, how to do the raw material testing to control the quality of raw materials coming in and even during your R&D stage. Okay. And actually, if you have online access, they quickly give you what kind of reference standards you need to buy when you want to do the testing. So they have listed down here. You need to buy USP reference standard or certain phosphate RS reference standard stand for RS, and then their related compounds RS, and one more is tributyl phosphine oxide RS reference standards. So this kind of reference standards need to be bought when you want to do the raw material testing, and also it involves in the regulatory requirement. I mentioned about 4-aminophenol just now for paracetamol, right? I also need to buy that 4-aminophenol reference standard because we are analyzing using HPLC where we need all these reference standards to analyze our products, our ingredients. Okay, for the ethical method validation, there is another document, okay, known as validation of ethical procedures, Q2R1. So these are other documents we need to put together and cross-check when you develop your drug delivery. Have all the parameters mentioned here for validation. Linearity, specificity, first, linearity, range, accuracy, precision, detection limit. If in the case of paracetamol honey just now, paracetamol is well established. We just need to verify the methods rather than validate the whole method and method. Right, and finally, these are what we come up with. Finished product specification. It is actually, we got it from the critical quality attributes just now. We will put nicely in what we call a document of a finished product specification, okay? So first parameter is appearance, identification using what kind of instrument, okay, and what is the spec, and what is the reference document that we use. For AC, we use LC, HPLC, and the spec is based on paracetamol suspension spec mentioned in USP here. This test. First performance test is the CQA, uniformity of dosage form, dosage unit. These are CQA we're talking about, and we already study what are the impact of manufacturing process against the COA, right? So we put them in the specification of finished product. So we already specified here what is the maximum volume, what is the minimum lower value, the upper limit, the lower limit, etc. Okay, based on USP here. And determination of density. There is, if not available, we need to do some in-house because these are the paracetamol honey, a novel formulation. Okay, so it is not mentioned in USP, paracetamol honey. But they mention paracetamol, we adopt the and do some in-house. And other performance test is deliverable volume. The average volume of liquid obtained from the 10 containers is not less than 100%. If we state there and claim 100 mil is the volume, it should be 100 mil, cannot be less than that for all the 10 that we sample out. Okay. In practice, for monophenol, we need to have this show evidence the chromatogram obtained in solution is this. Okay. And pH range, what is the pH range? And preservative assay, even the preservative assay is one of the ethical method that we need to develop. It's not only it is a requirement for registration for the suspension. It seems simple suspension, but we need to come up with preservative assay, method development, method analysis, and validation. If you put in 2 types of preservative, you need to come up with this 2 types. Or if you have a simultaneous detection that you develop, you can check it. Okay, it must fulfill the spec. And microbial contamination test. Okay, these are the specified total anaerobic microbial contamination, total yeast, specified organism test mentioned specifically for suspension is E. coli. Okay, so these are the requirements in USB. Okay, I come to the end of the lectures for today's session. I would like to conclude. If there's no questions, I would like to conclude my presentation with a few points. Okay. The design of drug delivery system shall be based on relevant guidance document so that we can ensure its acceptance. Data that we produce in the lab, in the wherever the stage of drug development, can be accepted by the regulatory, by the reviewer of that regulatory, by the member of experts before they can approve for registration. Okay. So if you follow the appropriate guidance, inshallah, you will it is easier to be marketed, inshallah, gain entry to the market. Okay, when we do the compiling all the data, all the testing, it should be recognized for the purpose of registration. The level of knowledge we gain is very important. We need to be knowledgeable with what we produce, with what the drug delivery system we are creating. Okay, we need to be knowledgeable. Some of the data is required for registration, some is not, but the data are still important. Okay, it provides the basis for scientific evidence or science-based submissions to fulfill all the regulatory requirements. So basically what we are doing in the lab, each single day in the lab you spend in the lab, you need to carefully observe whatever happening in your synthesis, in your mixing, even the cleaning of equipment. Okay, because this will be very useful when we do scale up. We need to give input on the cleaning process as well, because some of the materials, they can stuck easily to the glass apparatus. How you usually clean them in the lab will be very helpful to help people in the manufacturing later on. So every single day you spend in the lab, make it worthwhile. You need to observe carefully, and not only observe, you need to record them. Okay, make a good recording on your observation. Part of the observation can actually be used when you discuss your results data in your thesis. This is from our experience. All right, that's all. Wabillahi taufiq wal hidayah. Wassalamualaikum warahmatullahi wabarakatuh. I will pass back to our moderator.
Waalaikumsalam warahmatullahi wabarakatuh. Thanks, Professor Farahidah is completely talk about drug delivery design. But for me, our knowledge, this is very important, especially in halal. The halal enter to drug delivery design. It is very, very important. Okay, maybe any question from participant? In this chat, Prof. Farahidah, Mr. Rahmat Santoso, question. Can you explain the relationship between KBT and KBD? What is the design of experiment? And then others question, how is KBD implemented related to halal product in pharmaceutical industry? Perlu diketahui apa, Bu? perlu diketahui bahwa di Indonesia, Prof. Farahidah, kewajiban halal produk ini akan diwajibkan terutama untuk obat dan kosmetik ini nanti di tahun 2026.
Oh,
Karena
Masya Allah.
ya,
Then you really
ya, ya
need to
harus
list.
harus ada halal. Kalau
Yeah,
tidak
very
ada
good.
halal harus ditulisi non halal atau non halal
Oh,
begitu. But
okay.
untuk makanan untuk food sama beverage di
That
tahun
one
ini
already well established,
2024 ya,
right? Majlis Ulama
Oktober
Indonesia.
2024 harus sudah ada label halalnya. Oke, mungkin itu yang pertanyaan yang ada dulu, Prof. Mungkin bisa dijawab di
Yeah,
chat.
okay.
Oke.
Yeah, you mentioned about I'm getting to the chat. Cannot see the chat. So first question is about QBD.
PPT, yes.
Relationship between the quality by design with what? I didn't hear you. Sorry.
Relationship between KBT and KBD atau different KBT and KBD.
QBT. I actually
Yes,
have no idea about QBT. Quality by
quality
what?
by testing, quality by testing.
Quality by testing. Okay. So, quality by design, quality by testing, quality design built-in. These are all the names. One is that quality by design. When we design, we take care of the quality of the a few things, right? But with that quality is when what kind of tools we want to control the quality is by using testing, is by using certain tools. And that tools include RSM, include design of experiment, DOE, is part of the tools. Okay, but the tools is using or applicable at certain stage of drug delivery design, not all. If you are talking about, if you are researching antichemical method development, for example, antichemical method development, you are developing a method, right? You can employ DOE to do the optimization. Usually we do optimization, or prior to optimization, because it is a development, we are using Blackcat Burman software. It's a tools to help you to come to a faster conclusion, rather than prior knowledge too many. You need to do some ranking. If you are not satisfied with the ranking, you will do some tools using Blackcat Burman to narrow down what kind of factors you can select to narrow down your number of experiments, the final number of experiments. Okay. The quality by testing and quality by design. Okay. Quality by testing, I'm actually not really familiar, but what I can say, it could have a guidance that I can look to so that I can give you a better answer. But quality by design, what we have already employed in creating the pharmaceutical products is what I mentioned. Okay, it has a certain control. First, you need to start with the QTPP just now. It's a systematic development, and it needs to define a CQA need to be defined, and then you need to use tools. The tools is DOE software, and at certain stage you need to do optimization. Is a RSM, using RSM tools. So that are tools that are the framework. The QBD is a name that is a framework, and they come up with a guidance document for pharmaceutical, ICHQ8. For other engineering, I believe they also have their own terms. For accountant, they also have their own term. Okay, like the big term is for pharmacy is good manufacturing practice. Big term for financing, good financing practice. Big terms for agriculture, good agricultural practice. They also have their own guidance document. So that is my answer. The relationship between these all tools and QBD. What is the next question? I cannot retrieve the question at the chat box somehow. Maybe they just they message to.
How is quality by design implemented related to halal product in pharmaceutical
Oh
industry?
yeah. Okay. For halal, okay. QBD, the stage that involved is the quality of the raw materials, the critical raw material attributes. Okay. At that part, we need to specify what is the critical quality that we want. Say, for example, paracetamol honey just now, I want the halalness. I put it as the critical spec. So what in practice, we need to find manufacturer that provide halal certificate that can provide paracetamol with halal certificate, or they can provide paracetamol with process manufacturing flow, where from the process manufacturing flow, we can analyze what kind of doubtful materials involved in the processing of the paracetamol is there. This is one ingredient, right? Other ingredient that I use, honey, or other ingredient maybe stabilizer. So each of the maybe total 6 ingredients, I need to find the halal certificates of these 6 ingredients. I need to find manufacturers that can provide halal certificates. But don't be too worried, because for the pharmaceuticals and for many raw materials, especially for drugs materials, they don't really have halal certificate. What is the next thing to do if raw materials do not have halal certificate is to ask for process flow and to ask for halal declaration from the raw materials. So talking about how this QBD link with the halalness of raw materials is from the how we control the critical material attributes just now. One of the spec is halal. That is from the halal parts. We don't really need to test whether there is gene of porcine contaminate our raw materials or not. We don't really need to test that. Okay, but it is suffice if we can provide justification to the halal certification auditors. We just mentioned our paracetamol is halal based on these certain documents. These are what SOP we practice. You need to have a certain SOP, how you evaluate the halalness of your raw materials. I can give one topic, one lecture on halal evaluation of raw materials later, inshallah.
Thank you, Professor.
Okay, thank you. Okay, thank you, Professor. Maybe any participant direct question? Other participant? Jadi implementasi halal di produk ini tadi disampaikan Profesor Farahidah, terutama di critical material atributnya. Jadi materialnya harus bersertifikat halal begitu, dan tidak boleh mengandung sesuatu yang haram. Nah, kita cukup sebetulnya tinggal ketika pembelian bahan atau sumber bahan yang kita cari adalah yang mempunyai atau ada sertifikat halalnya. Itu salah satu implementasi KBD halalnya gitu. Seperti
And
itu.
of course, just to add, on top of halal certificate, if they don't have, it's okay. But all of us need to be very competent on analyzing based on nature and origin of the raw materials.
Yes.
Say for example, silica, or if you use talc, TALC.
Water, maybe water.
Water is
Water.
important.
How about process, water process? Yes.
Yeah, in the
Filter of
processing
water.
of
Yes.
in the
Filter.
process of paracetamol,
Yeah.
pharmaceutical water, there is filtration system, right?
Yes.
In the filtration system, you need to study what is the material in the composition of the materials in the filtration system, water purification system. Kita panggil dalam factory lah. Dalam industry, we call it as WPS. Engineering term dia WPS lah kita panggil as one of the facility, as one of the utilities in JMP. We need to have this WPS, water purification system. So one of the filtration mechanism is the membrane, right? So that membrane, if as much as possible, if you are a factory that would like to apply for halal, a certificate for your products pharmaceutical, you need to have certificate of halal for the WPS. The membrane itself needs to be certified halal, or it can you can ask the supplier to declare what is the source of the membranes. Is it from membrane, and also the carbon, the activated carbon. It's very important because carbon can be from bone of animals, can be from woods materials. So that is halal for the water in pharmaceutical.
Oke, oke. Begitu ya, Mas Rahmat Santoso. Gimana, Bu?
Terima kasih.
Bu Nur, sudah? If any question again, it is enough and very clear and completely. Maybe this lesson in morning we can diakhiri apa, Bu? We can close, and inshallah Miss Nia will be send your certificate to you via email, ya. Okay, maybe Mrs. Professor Nur Hasanah ceremonial closing in this morning, this lesson.
Okay. Thank you very much, Prof. Farahidah, for your interesting lecture this morning. Yeah, very beautiful. And we will give you a certificate, and yeah, this is the certificate which will be delivered to you through email. Thank you very much for your lecture today.
Thank you very much.
And hopefully we can meet at the May, and you can attend the Faculty of Pharmacy Universitas Ahmad Yani. Okay, thank you very much.
Inshallah. Thank you very much for inviting me to share some of my experience. Alhamdulillah, hopefully, as mentioned by our moderator, there is a need for halal and then maybe being enforced in Indonesia soon. So 2 years, maybe 2 years, but in 2 years time, right?
Yes, yes, yes.
In 2 years time, you
Yes.
prepare.
2 years for cosmetic, but for medicine, I think the
26, 2 tahun.
29.
Ya, ya.
Okay.
26.
Traditional medicine 26, but for medicine 29. Chemical medicine 29.
But the preparation for 2029, I believe, need to start from now.
Yeah.
Because they need to renew and they need to do some.
Very close. Yeah.
Okay, inshallah.
Oke, terima kasih kepada Profesor Farahidah dan para partisipan semua. Marilah pertemuan pada siang hari ini kita tutup dengan membaca hamdalah dan doa kifarah majelis bersama-sama.
Alhamdulillahirrahmanirrahim.
Subhanakallahumma
wa bihamdika asyhadu an la ilaha illa anta astaghfiruka wa atubu ilaik.
Thank you, Professor Farahidah. And next, see you next in May.
On May.
Insyaallah.
Yeah.
Inshallah.
Terima kasih. Assalamualaikum warahmatullahi wabarakatuh. Apabila ada
Waalaikumsalam.
kesalahan saya minta maaf.
Selamat menyambut Lebaran.
Terima kasih.
Maaf lahir batin.
Ya, terima kasih. Terima kasih.
Prof. Farahidah, boleh kita dapat materinya nanti?
Materialnya. Okey. Allah. Boleh.
Boleh, nanti boleh share ke atau boleh di-share di chat, room chat?
Oh, chat here, you mean?
Atau ke Pak Mustafa juga?
Ke Pak tu ya, yang live dengan saya tu?
Ya, Pak Mustafa.
Pak Mustafa, ya. Okay, insyaAllah. Right.
Ya, terima kasih. Terima kasih. Waalaikumsalam.